Hello, everyone. I am Palanisamy from the Henry Ford Health System in Detroit, Michigan, United States. First, I would like to thank the organizers of this meeting, particularly Select Science, for giving me this opportunity to present my work on prostate cancer biomarker discovery. So today, I'll be talking to you about the different biomarkers that I was involved in the discovery, and I will share with you some of the insights, how we made those discoveries and what are the potential applications of these new biomarkers in prostate cancer. So one quick slide, give a definition. What is a cancer biomarker? A cancer biomarker is a measurable substance or a molecule or a characteristic feature that provides information about cancer's presence, its progression, or response to treatment. So these biomarkers are often derived from tumor cells or the body's response to the tumor and can be found in blood, urine, tissue, or other body fluids. So there are different categories of biomarkers. They could be diagnostic, prognostic, predictive biomarkers, therapeutic biomarkers, or some of the biomarkers are used for monitoring the treatment response. So, for example, in prostate cancer, the prostate-specific antigen is routinely used for the diagnosis, and the BRCA1, BRCA2 mutations, although all the patients who carry the mutations may not develop the breast cancer, but these markers are used to assess the risk of developing breast cancer. And the predictive biomarker is the HER2 overexpression in breast cancer that predicts the responsiveness to the treatment to the drug trastuzumab. And for immunotherapy, the PD-L1 expression is used to find out the efficacy of whether the patients are eligible for immunotherapy. And similarly, the CA125 marker is routinely used for initial diagnosis as well as the monitoring the treatment response. So there is largely different categories of biomarkers. So this topic is going to be mainly on prostate cancer. So I'm going to share with you some of the work that I have done in my laboratory. So before going into the details, just one slide to show the global incidence of cancer, overall all different type of cancer. So every year, at least about 20 million people are diagnosed with a different type of cancer. So almost half of them are in males and half of them are in females. And if you look at the prostate cancer alone, there is about 1.4 million new cases are diagnosed every year globally. So in the United States, there are about 300 new cases are estimated to be diagnosed in the year 2024, and of which about 35,000 people may die with prostate cancer. So it shows the importance of actually doing in-depth studies to understand the way to find a cure for this cancer. So the prostate cancer in general, it takes about 10 or 20 years in the making. It goes through a lot of process from a normal epithelium to a metastatic cancer. In general, prostate cancer are treated with the androgen deprivation therapy because prostate being a hormone-driven cancer, if you suppress the androgen, they give a good response. So some of the patients, they may not respond to the androgen deprivation therapy. They develop resistance to that. So those cases are called castration-resistant prostate cancer, and some of them may differentiate into neuroendocrine prostate or metastatic prostate cancer. So there is so much to understand the genetic complexity of prostate cancer from early stage to the advanced stage. So the early stage prostate cancer, initially it was diagnosed with the elevated PSA, that being a biomarker. Although the PSA is not specific for cancer, but that is the only best available marker for initial diagnosis of prostate cancer. So then Initial diagnosis, the patients will get a biopsy diagnosis, and the pathologist will examine the prostate based on the morphology. They can classify into different Gleason grades, Gleason from early stage to the advanced stage, like Gleason 3+3, 6, or 3+4, 3+7, or up to like 5+5, the higher grade. So when you go to the metastatic cancer, they are not heterogeneous. They are kind of clonally identical. All cell population may be the same. You may not see different kind of Gleason pattern. So the main focus of our research to identify what are the genetic changes that occurs during the growth of this prostate cancer from early stage to metastatic stage. So here I'm showing you one prostate, the cross section of the entire prostate gland. So the main thing that we identify in most of the prostate cancer is the cancer is a multifocal cancer, meaning that there are many tumor foci will be present within a prostate. So there is an area that is circled in green. This is a large tumor. And there are other smaller foci that are circled in yellow and other colors. So each marker, so each tumor foci are genetically distinct, which I'm going to talk more in detail later in the presentation. So this shows the enormous heterogeneity of the prostate cancer. So prostate cancer is not a single cancer. There are cancer within cancer, multiple cancers are there. So what we need to find out is the inter and intra-tumor heterogeneity at the molecular level. So I will talk more in detail in the subsequent slides. So since we have this enormous heterogeneity of this prostate cancer, our challenge is to identify, are there any distinct biomarkers that we can identify? So I will show you the discovery of different cancer biomarkers. So as we know, more than like 80% of the epithelial cancers are present in all different organs. And only about 10% of the cancers are like leukemia or lymphomas. So the one well-established biomarker in cancer is a gene fusion, meaning that there are two different genes located in two different locations. They undergo translocation, or they come together by genomic deletion, like that. They form a new combination of two genes. This is called fusion genes, and the overexpression of the fusion gene is responsible for the cancer development. So like that, the TMPRSS2 ERG gene fusion was first identified in any of the solid cancer. Before that, we know the existence of gene fusion in leukemias and lymphomas, like chronic myeloid leukemia, BCR-ABL, like that. But in a solid cancer, and the discovery of the TMPRSS2 ERG is the first major discovery. It was made by analyzing the cancer outlier profile analysis method. By combining the different microarray data, we are looking for the gene that is always highly expressed. So in prostate cancer, by combining the multiple microarray data set, we found that ERG is the highly overexpressed gene. And when we look at the transcript of the ERG, what we found was it is not a full-length transcript. The entire ERG gene was not expressed. Only from exon up to exon 1 to 4 is not expressed at all. And then from exon 5 onwards, the rest of the gene is expressed. So this imbalance in the expression of the RNA transcript clearly shows that it is a fusion gene. So by doing the race PCR, what is called rapid amplification of cDNA and 5' race CR, 5' race PCR from exon 5, that help to identify the 5' partner, which is the TMPRSS2 ERG. So the first exon of the TMPRSS2 is fused with the exon 4 of the ERK gene, which results in the overexpression. So this was confirmed by fluorescent in situ hybridization analysis. The fusion event was confirmed. So this is the structure of the fusion gene, the TMPRSS2 ERK gene. The TMPRSS2 is an androgen responsive gene. So the androgen receptor can bind to the promoter of the TMPRSS2, which drives the overexpression of the ERK or other X gene fusions have been identified, which I will show in the next slide. So prostate being an androgen-regulated gene, androgen-regulated cancer. So the gene fusion uses a gene that is responsive to the androgen receptor. So that drives the overexpression of the ERK. So after the discovery of the ERK gene fusion, there are other EDS family genes like ETB1 and ETB4 also identified in prostate cancer. The ERG, ETB1, ETB4 are a family of ETS family genes or E26 transcription factor. There are about 26 genes within the family. So within that, these three genes are expressed in prostate cancer. So it is important to note that this gene fusion, the ERG, ETB1, ETB4, they are not expressed in benign prostate at all. Only after the gene fusion or the translocation event, these gene fusions or these genes get overexpressed. So which clearly shows the specificity of these gene fusions in prostate cancer. Then after the discovery of the EDS gene fusion, we also identified another gene called SPINK1, which is called serine peptidase inhibitor result type 1. So this is not a gene fusion. This gene is expressed only the normal gene is overexpressed only in prostate cancer tissues, but not in the benign at all. So this is we also show that since there is not a gene fusion, we showed that the epigenetic silencing of the microRNA 338 and the 421 is responsible for the overexpression of the SPINK1. And also we showed that androgen deprivation upregulates the SPINK1 expression and also involved in the cellular plasticity. And we also reported the integrated proteomic approach. We identified a tyrosine kinase kit as a therapeutic target for SPINK1 positive cancer. So these markers are, these biomarkers are very important in prostate cancer. And also we show that the SPINK1 overexpression is associated with the worst recurrence-free survival. As you can see, the SPINK1 positive cancer patients do worse when compared to the SPINK1 negative cases. So, and also we show that the SPINK1 expression is regulated by the androgen receptor. So whenever the androgen receptor is expressed, the SPINK1 level goes down. The AR positive tumors, they do not express SPINK1, whereas if the androgen receptor is negative, you will see the SPINK1 overexpression. So that is how much inverse correlation of the androgen receptor versus the SPINK1 expression. So with the discovery of the ERG, ETB1, ETB4, and SPINK1, all these markers, they are not present in all the tumor patients, okay? They account, all these 4 genes account for about 50 to 60% of the prostate cancer. So the remaining 40%, we don't know what are the gene fusions or any other genetic mutation. So in order to find out, are there any new biomarkers that we can discover in the EDS fusion and the SPINK1 negative tumors, we use the next generation sequencing technology. As you all familiar, next gen sequencing technology is a powerful approach for in cancer research. One can do several things, like you can do DNA sequencing, RNA sequencing, or chip sequencing, or target capture, or resequencing. So for our purpose, we focused only on the transcriptome sequencing, meaning that we sequence only the messenger RNA. So once you get the whole transcriptome sequencing, one can do several type of analysis, like you can look for gene expression, SNP discovery, fusion transcripts, viral genome, pseudogenes. So our research is focused on looking at the gene fusions And also we looked at the pseudogene. That's another class of non-coding RNA, which I will show in the subsequent part. So for those who are not familiar with the next-gen sequencing technology, here I will show you one slide. So basically, when you are doing the messenger RNA or the transcriptome sequencing, you have to isolate the total RNA from the tumor and select the polyadenylated RNA transcripts, and you fragment them to 300 or 500 base pairs. Then we put adapters at both ends. These adapters are put it in the sequencing machine. So once you then the sequence only from the ends of each DNA fragment. You don't have to sequence the entire fragment. So just 300. Depending on the time and the depth sequencing, you can get up to like 150 or 200 base pair reads from each end. So once you get the sequence, these are all called mate pairs. So by using bioinformatics, you can actually combine the 5' and 3' end sequence of each fragment. Then they call it as mate pairs, and these mate pairs are mapped to the normal reference genome, so that we know exactly where they are mapping. So depending on the number of mate pairs located in a particular location, you can assess the expression level. And some of the mate pairs, if they are involved in the fusion gene, they may not map to the normal reference genome. So those mate pairs will be analyzed separately to discover the gene fusion. So using that approach, what I did was I carefully selected cases that are negative for H gene fusion and SPINK1, and we subjected them for next generation sequencing. We sequenced about 25 prostate cancer patients, and of which one of them we found that very high level expression of a gene called BRAF. This is a very important gene, very frequently mutated in melanoma. But this first time we showed the high level expression of the BRAF in prostate cancer. Then we selected this patient and we want to characterize, like we did for the ERG gene fusion. We did the exon spanning, using exon spanning primers. We did the PCR, and what we found was only from exon 8 onwards, we see the high level expression of the transcript, but the exon 1 to 7, there was no expression. As you can see at the bottom, this is the mapping of the sequence reads to the BRAF gene. Only from exon 8 onwards, we see the high level expression of the transcripts, but there was no expression from 1 to 7. So this clearly shows that this is a fusion transcript. Then since we have the mate pair reads, we know the 5', the end of the sequence as well. So what we found was the other gene that is fused with the BRAF is the SLC45A3. So the first exon of the SLC45A3, which is again is an androgen regulated gene that is fused with the exon 8 of the BRAF, which results in the overexpression of the BRAF. And also we found another family of the BRAF RAF kinase family genes. The RAF1 is also, we found on another case, RAF1 also involved in the fusion, which we confirmed by using the fluorescent in situ hybridization. We confirmed the rearrangement by fusion and break apart probes. So this clearly shows just from the next generation sequencing, we were able to discover a new biomarker. So after our report of this discovery, there are several other cancer types, other solid cancer also found to have this BRAF gene fusion, BRAF and RAF1. It is not only in prostate cancer, it was identified in brain cancer, pancreatic, gastric, thyroid, skin cancers. So one important thing to note is that in all the solid tumors, the BRAF gene fusion, it retains these the 3' tyrosine kinase domain, which is the kind of the functional part of this fusion. And for each cancer type, the 5' partner is completely different because essentially it is using the tissue-specific promoters from each cancer type. So the 3' end of this fusion is the functional unit, which drives the overexpression and responsible for cancer development. So now I showed you that we discovered ERG ETV1, ETV4, SPINK1, and BRAF and RAF1. So these are the 4 or 6 genes they are serving as a biomarker in prostate cancer. So when we analyze the next-gen sequencing to identify if any other genes are involved where we can find any new mutation, we could not find any new mutations. Therefore, since we have a large compendium of sequencing data, we focused our attention looking at the non-coding RNA landscape. So recently it has been shown that non-coding RNAs are expressed specifically in a cancer-specific manner. Some of them are in normal tissue and some of the non-coding RNAs are expressed only in cancer type. So they play an important role in the regulation of many genes. So our focus was mainly on one class of not long non-coding RNA. We focused the attention to look at the pseudogenes as a new family of non-coding RNA, because the pseudogenes were thought that they are junk DNA. It was found to be later, it was not, originally it was thought as junk DNA, but now we know that they are actually functional part of the human genome. So the pseudogenes are originally discovered in the 1977 by Jack et al. And they are basically the dysfunctional relatives of known genes that have lost their protein coding ability or not expressed at all. So any genomic sequence that is similar to another gene and defective is called a pseudogene. So there are different types of pseudogenes called processed pseudogenes or non-processed pseudogenes and disabled pseudogenes. So in the essence of time, I may not be able to go into the details. So I encourage the readers to read more about these different categories of pseudogenes. So what we did was, when we carefully collected the pseudogene specific sequences from our next-gen sequencing data, we clearly show that some of the pseudogenes are expressed in a tissue-specific manner, and some of the pseudogenes are expressed in a cancer-specific manner. Okay, so and there are many many pseudogenes have been identified in the human genome, but in prostate cancer we identified about 7 different pseudogenes that are expressed in a cancer-specific manner. So interestingly, in our pseudogene analysis, we found an interesting gene fusion. Since our focus was to discover new biomarker, we looked at the expression pattern of all the pseudogenes, and interestingly, we found one gene fusion involving a gene called KLK4 and KRSP1 or KLKP1. It has both names. So what we found was the first 2 exons of the KLK4 is fused with the exon 4 and 5 of the KLKP1. So a KLKP1 being a pseudogene, it is not expressed, it is highly expressed, but it is not coding for any specific protein being an uncoding RNA. So interestingly, what we found was with the result of the fusion and the change in the open reading frame, part of the pseudogene after the fusion is converted into a protein coding gene. So this is the first evidence that we show in prostate cancer, a non-coding pseudogene is converted into a protein coding genes with the translocation. So the bottom, the amino acid sequences shown in blue are from the KLK4 protein coding gene, and the amino acids in the red are the new set of amino acids that is coming from the pseudogene. So we designed a fusion-specific primer for KLK4 and KLKP1, and we did a PCR, and we showed that the expression of the KLKP1 only in prostate tissue, not very high in any of the prostate cancer cell lines. It is not expressed in benign prostate or any other solid cancer. So which clearly shows that the KLK4 KLKP1 fusion is enriched or specific for prostate cancer. To further validate our finding, we looked at the GTEx portal, where we have 54 normal tissues, across 1,000 individuals have been sequenced, and these sequencing data is available in the GTEx portal. Anyone can look at it. So what we found was the KLKP1 is expressed only in the prostate tissue and not in any other another 53 different type of tissues, the normal tissues in the human. So what it says that the normal KLKP1 transcript is specific to prostate, but the KLK4 KLKP1 is expressed only in the prostate cancer tissue. So which clearly emphasize the specificity of this KLKP1 for prostate cancer. So this is yet another new biomarker that we discovered. Then here is the complete genomic organization of the gene fusion, KLK4 fusion with the KLKP1. And this is the full-length RNA sequence. And this is the peptide sequence of the 165 amino acids. And we developed an antibody, and we showed that it is expressing a fusion protein. And since it is a new protein, we don't have a good antibody. That's why we use the RNA in situ hybridization or RNA scope to study the tissue level expression of these KLK4, KLKP1 fusion genes. And we clearly showed that only in the high Gleason grade, the high level expression of this fusion gene. And also we found that this fusion is expressed in prostate cancer in young men than the older age patients. Patients who are less than 50, age 50, we found this fusion. So this shows the, association of this fusion in the young men with prostate cancer. And in all these cases, the PTEN, which is another biomarker, which is commonly deleted in prostate cancer, but this fusion is expressed in the prostate tissue that is with the intact PTEN. So for more details, please refer to this paper. We already published this paper in 2019. So far, we clearly show that the discovery of ERG, ETB1, ETB4, SPINK1, BRAF, RAF1, and KLK4, KLKP1. Which clearly shows that the presence of these prostate cancer specific biomarkers. So now you can see these are the different all these different biomarkers that are discovered in prostate cancer. So the one area that I did not talk more about this, another non-coding RNA is called PCA3, that is known to be expressed in about more than 90% of the prostate cancer. Then PCAT1, SLAP1, these are the two other non-coding RNAs. Also, these are long non-coding RNAs, not pseudogenes. They're also expressed in prostate cancer. As you can see, we have a long list of these biomarkers that are specific for prostate cancer. And the detection methods are varying, because if you have a good antibody, you can actually detect them by immunohistochemistry. And if you don't have a good antibody, we can do by RNA in situ hybridization. And again, non-coding RNA and pseudogenes can also be identified by RNA in situ hybridization. So with all the discovery of all these biomarkers, this is the first time we were able to classify the prostate cancer into distinct molecular subtypes, where you have the ERS gene fusions, ERG, ETB1, ETB4, and there is another gene, ETB5 also there. Then we have a SPINK1 subtype and the RAF1 and the KLKP1. So what is interesting is these biomarkers, although the ERS family genes are like transcription factors, unlike enzymes, they are very difficult to target. That's why there are many studies are going on, they can be indirectly targeted ERG positive tumors can be treated with PARP inhibitors. And there are some clinical trials going on with that. And some small molecule inhibitors are developed for the ETB1, and the SPINK1 can be targeted with the EGF inhibitors, and the RAF gene fusions can be targeted with the RAF or the MEK inhibitors that we showed in our paper. So this clearly shows that these biomarkers, not only we discovered the biomarkers, there are actually therapeutic potential or therapeutic possibilities for these biomarkers. So that's why this becomes clinically more important. So once you have a tissue biopsy, we can screen these all the biopsy tissues with all different known biomarkers, and you can select the specific inhibitors, and you'll be able to treat these patients based on the specific inhibitors. So this clearly shows the potential clinical application of these biomarkers that we discovered. So sometimes the patient may not respond to the treatments that is specific to the particular markers. Then we have to rebiopsy them. Then we have to do some next-gen sequencing to identify some new targetable biomarkers. So, because of the availability of these different biomarkers, we can classify or stratify the patient into distinct molecular subtypes, and so that we can offer like a personalized medicine. And so, based on the responders versus the non-responders. So, therefore, so the screening of the biopsy tissue, whether by PCR, FISH, or IHC or RNA-H, need to be done routinely to identify these markers. So having discovered, so this is not straightforward, okay? Although we have these many markers, we have the therapeutic possibilities, still there are actually a lot of challenges. What are the challenges? Like I showed you in the hormone tissue, where the prostate is a multifocal disease. There are multiple tumors within the prostate. It's called the tumor heterogeneity. So the genetic diversity within and between the tumor complicates the biomarker validation, and the single biomarkers may not capture the complexity of the cancer biology. If you are screening this prostate with one biomarker, you conveniently miss a lot of information. So that's why there is a big gap in the translation of this discovery from the bench to the bedside. And also the cost of the biomarker test limit the access, and especially in the low risk settings like that. So I will talk about the challenges with the biomarker. So like I said before, the prostate is not a single disease. It is a multifocal disease. More than 90% of the prostate cancer patients having this multifocal disease, meaning that at least 3 or 4 different tumors at different stages of development. You will see it in the entire prostate. So it could be the high-grade PIN or the adenocarcinoma, or low-grade tumors or high-grade tumors. You will see all different type of cancer within a patient. So our challenge is to identify, like in this example, I'm showing the entire cross-section of the entire prostate, where we see the 5 different tumor foci. Okay, so when we screen this tumor using multiplex IHC and RNA scope, we screened with like a SPINK1, ETV1, and ERG. What we found was, interestingly, like one tumor foci is positive for ERG, other is positive for SPINK1, and the third foci is positive for ETV1. So this is the first time we clearly show that in a solid cancer, that more than one gene fusion is present within a patient. So and the bottom circle, this is the largest tumor, the so-called the index or the dominant foci, that is completely negative for any of these markers. So conventionally, most of the clinicians and pathologists, they always take the dominant or index foci, and they do this biomarker analysis, and they call it major diagnosis. So just by analyzing only one tumor, we are conveniently missing a lot of important information. So that clearly shows the enormous heterogeneity of this prostate. So therefore, instead of using only one of the tumor foci, I would encourage to do a whole mount. And we have to do an unbiased screening of the prostate cancer to evaluate these biomarkers, because each tumor carry entirely a different biomarker, and there is no commonality. Since each tumor are developing individual at different time, they carry different driver mutations. Okay, so this is what I want to show you more in depth. So this is entirely the whole mount prostate, where we stained with both ERG and SPINK1. The region that is stained in blue is the SPINK1. There are some areas they are stained with the brown, that is called ERG. As you can see, the enormous heterogeneity of this tumor. Look at this, the SPINK1 expression pattern. So there is a big range from high-level expression to low-level expression. Then in between, within the SPINK positive area, there is another tumor foci that is positive for ERG. So this clearly shows that each tumor foci is evolving independently with a different driver molecular aberration, which shows the heterogeneity. So if you take a biopsy or any sampling of this tissue, you're going to make a punch from only a small area. If you evaluate that, you will conveniently miss a lot of information. So therefore, I insist that we have to do an unbiased evaluation using the whole mount prostate. So here again, I want to show you the zoom-in view, where you can see the different level of expression of the SPINK1 in blue. And within that, there is a high-grade PIN that is positive for the ERG gene fusion. This clearly shows the enormous heterogeneity of the prostate cancer. So we did more than 800 cases like this, the whole mount we analyzed with these biomarkers. What we found was there are cases, good number of cases that are positive for like ERG-SPINK ETV1, the 3 marker positive, and some cases are actually even positive for all 4 markers and all 4 markers. So when they are doing the radical prostatectomy, the multiple tissue blocks have been identified. So one has to screen all these blocks to capture like every foci with the marker. Okay, so this clearly shows the extent of this complex tumor molecular heterogeneity of prostate cancer. So also, it is not only that we discovered this biomarker and clinical application, but we found that these markers are associated with clinical outcome. Okay, so we studied these markers in a racial disparity perspective using Caucasian and African American. What we found was the ETV4 positive patients are associated with worst recurrence-free survival, particularly in African American, whereas the ETV1 expression is associated with the worst recurrence-free survival in Caucasian Americans. So this is a new data that we recently is unpublished data. So this clearly shows these biomarkers have an important kind of role in prostate cancer development, and they have a utilization for early diagnosis as well to assess the recurrence risk. So this we can get this information only by doing an unbiased analysis of the anterior prostate. Either you can do it on the whole mount, or we can use the saturation biopsy samples where more than 24 cores are pulled out from the prostate. And when we screen all the tumor foci carrying cancer, we will be able to identify either one or more of the sets gene fusions in prostate cancer, and then we will be able to intervene with the treatment much earlier rather than doing watchful waiting. So now I think the next few minutes I will show you, having identified all these biomarkers in prostate cancer, I didn't have time to show a slide where we can show that these gene fusions like the TMPRSS to ERG, the PCA3, and the KLK4, KLKP1, all these gene fusions can be identified in the urine of the prostate cancer. Even before doing the biopsy, when the patients, they have elevated PSA, we can collect the urine, we can detect these gene fusions in the urine itself to confirm the cancer diagnosis. Then after that, either they can go for biopsy or directly surgery or hormone treatment like that. So the other advantage of having this biomarker is we can capture these biomarkers in the circulating tumor cells, because sometimes the patients, when they have high volume disease, you will see these cells circulating in the blood. We can capture these circulating tumor cells by using different methods, but we use what is called the microfluidic device. We take about 2 cc of blood and pass through this microfluidic device. When you pass through the microfluidic device, as you can see, there are multiple wells in the microfluidic device. In our system, we had about 56,000 small wells. At the bottom of the well, there is a small hole that is small enough to allow, or large enough to allow only the WBC and RBC, but not the epithelial cells or the cancer cell. They are much larger. So they get stuck in the well. So we can do the immunohistochemistry for the cytokeratin and the CD45, that is cytokeratin specific for epithelial cells, CD45 for the white blood cells. So we can distinguish between the epithelial and the other white blood cells. So in this way, we can capture the circulating tumor cells. Then we can isolate the circulating tumor cells. Then we extract the DNA and RNA. You can subject them by PCR or sequencing methods. You can detect these gene fusions in the circulating tumor cells. So these markers can be used for early detection. So recently, there is another approach called cell-free DNA, or the circulating tumor DNA can also be captured. These are all small fragments of the DNA, not encapsulated in a cell, but the DNA are floating in the circulating blood. So we can capture those DNA, and by analyzing those DNA, we can actually identify these gene fusions and the biomarkers. So from the marker discovery, I showed you the clinical applications and the discovery and actually screening in the urine and association with the cancer clinical outcome and the potential for early detection of the prostate cancer. So in summary, I want to show you that with the discovery of several new biomarkers that led to the molecular classification of the prostate cancer, and I showed you that using the hormone prostatectomy tissues, which revealed an unprecedented details of tumor heterogeneity with respect to molecular markers, clinical and pathological parameters. So ours is the first study reporting the incidence of multiple molecular markers in a large cohort of PCA from racial disparity perspective. Because you don't see this presence of multiple gene fusion in any other solid cancer. Like even in lung cancer, there is a ALK gene fusion, ROS, RET, MET, and different gene fusions are there. In lung cancer, we don't find more than one gene fusion in any given patient. There may be some rare exceptions, but generally you don't see it. But in prostate, we are seeing a high frequency or a high incidence of these multiple molecular markers. So our study is significantly different from other previous study that we have evaluated the whole prostate tissue instead of the dominant tumor nodule. Okay, there is no unbiased. Our approach is totally unbiased. So the distinct molecular marker incidence in separate foci suggesting independent clonal evolution of tumor in a multifocal disease. So given that more than like 90% of the PCA cases are multifocal, our study results emphasize the importance of considering the secondary nodules when making treatment decisions, not only the dominant or the index nodule. So the distinct molecular subtypes of pathological differences between Caucasian and African American patients reaffirms the racial disparity among PCA cases. So what we show is the molecular heterogeneity and racial disparity could be a major factor that dictate the overall response to the treatment options and will considerably impact targeted approaches for PCA management. Because since we have these multiple markers present within the patient, even if you have drug for one marker, it will work only on that tumor foci and the other tumor foci will still continue to grow. So this is the biggest challenge. The tumor heterogeneity is a major problem where we are not able to find effective cure for that. So all these observations are made on the localized prostate cancer. Okay, when you go to the metastatic, there may be heterogeneity between different metastatic sites. But otherwise, each metastatic tumor will be clonally distinct. They may be just the uniform cell populations. You may not see heterogeneity within the metastatic site, but with the different, like visceral and the bone metastasis, you may see some differences in the market. I think there are studies, they showed the heterogeneity only between different metastatic sites, but not within the same metastatic. So all these aberrations or the mutations are captured in a localized prostate cancer. So this is what we are trying to show with the identification of different molecular markers, whether in prostate or lung cancer, we can classify the cancer into distinct molecular subtypes and develop some drugs targeting based on the biomarkers and which will help the patients for treatment benefits. Okay, even because this is completely a personalized approach, because since these biomarkers, whatever we discovered, they are not present in 100% of the cancer patients, it identifies only distinct subtypes. Only small subsets of each biomarkers can be identified. Therefore, we have to do an unbiased screening of the tumor to capture all the known biomarkers and help the patients with the better treatment options. So with that, I would like to thank, this is many people's effort, not only me. There's a lot of collaborations going on, and especially I want to thank particularly Shannon Koskaden. She is our research coordinator in my lab, she did all the pathology work for screening biomarkers and the tissues, and of course several other clinicians, oncologists, and several other collaborators who helped for the success of the study. And also I want to thank the funding support from the different funding agencies. So with that, I would like to conclude these presentations and I will be happy to answer any questions. Thank you very much.